Okay, so welcome back to this next video in which we are discussing heart failure and heart failure medication. Okay, right, so we're currently in the process of discussing hypertension-induced heart failure. Okay, and what we have seen is that if you have chronic arterial hypertension, then what it's going to do is it's going to make it more difficult for the heart to pump blood into the arterial system, and this is going to cause the stroke volume to decrease, okay, which will then lead to cardiac output decreasing. However, there are all these neurohormonal mechanisms in place that are going to mean that we're not actually going to end up decreasing mean arterial pressure. Okay, you would think that because cardiac output has gone down, mean arterial pressure will go down. However, there are so many neurohormonal mechanisms for controlling mean arterial pressure that arterial pressure isn't actually going to go down. Okay, so that's going to remain roughly the same as where we've got to so far. In addition, to maintain normal mean arterial pressure, the sympathetic nervous system is going to be chronically on, okay, and it's going to be stimulating the heart to beat harder and also to beat more rapidly. So the heart rate is chronically too fast, okay, so you get a chronic tachycardia. In addition, we've discussed that the central venous pressure and the pulmonary venous pressure are going to be chronically increased because of the increase in blood volume. Another thing that I would just like to add on to this little list of points that summarize what is going to now happen is that angiotensin II is going to be chronically high, okay? And all of these things together are now the perfect breeding ground for heart failure, basically. They're going to trigger cardiac hypertrophy, basically. This is the next thing that we now need to discuss, hypertrophy. Okay, so these things are going to lead to what's known as ventricular remodeling, okay, or remodeling of the heart. Okay, now the main feature of remodeling of the heart is hypertrophy of the cardiomyocytes, so that's what we're going to concentrate most on. Okay, right, so the cardiomyocytes are going to be stimulated firstly chronically by the sympathetic nervous system and also by angiotensin II. So that's the first thing which we'll talk about because that's the simpler thing to understand. Okay, actually, before I talk about that, I think what I'll talk about is I'll talk about what hypertrophy actually means, okay, and then we'll come back to discussing how these four things here, mean arterial pressure being too high, chronic sympathetic stimulation, too high central venous pressure and pulmonary venous pressure, and chronically elevated angiotensin II levels are going to cause hypertrophy. So, what does hypertrophy mean? Well, hypertrophy means that the cells of the heart, the cardiomyocytes, are actually going to grow. Okay, so basically there are two ways that any tissue can grow. So if I show you a little picture here. So let's say that this is a tissue, this collection of cells that I'm drawing here. So here is some little tissue here made up out of this collection of cells. So if you want to grow a tissue, if you want the tissue to get bigger, there are two ways that it can get bigger. The first is that the individual cells could get bigger, okay, like so. So all of these four cells have got bigger now. Okay, that is what is meant by hypertrophy. The cells have actually got bigger. So it's a way of growing a tissue but without actually increasing the number of cells you have. Okay, and the way around having to increase the number of cells you have is just to grow the existing cells, make the cells that you already have bigger. Okay, so that's what hypertrophy means. It means that the cells are actually going to get bigger. Okay, the other way that you could grow a tissue is that you could actually make more cells. Okay, so the other way is that the cells could remain the same size, okay, but now you're going to increase the number of cells. Okay, so I'll increase it to 9 maybe, and that's roughly the same size as the hypertrophy tissue up there now. Okay, so this is another form of growth, okay, involving cell division, and this is known as hyperplasia. So the two main forms of growth that are available for a tissue are hypertrophy, where the actual cells themselves are going to grow, and then hyperplasia, which is where the cells are going to divide, and then that's also going to increase the size of the tissue, simply because if you have more cells, it's now going to be a bigger tissue. Okay, right. Now, the heart is not capable of hyperplasia. Okay, you cannot grow the heart by having more cells produced, okay? Cardiomyocytes cannot divide, okay? The cardiomyocytes that you have in your heart now are the same cardiomyocytes that you had when you were born, okay? The cells just can't divide. They don't have that ability, okay? So if you want the heart to grow, the only way you can actually grow the heart is through hypertrophy, okay? The cardiomyocytes individually get bigger, okay? And this is what this combination of four things for an extended period of time is going to do. It's going to make the cardiomyocytes grow, okay? Now, this is done as a compensatory mechanism, okay? The heart isn't failing, basically. It's weak. It's failing to pump enough blood into the arterial system because of the too high arterial blood pressure. Okay, so what we can do is try and strengthen the heart. Let's make all of the cells bigger so that their contractile ability gets bigger. Okay, now there are two main different forms of hypertrophy, okay, that can occur. One is concentric hypertrophy and the other is eccentric hypertrophy, and I just want to make sure I explain what the difference between these two is. So if this represents a cardiomyocyte here, so let's say this is a cardiomyocyte, okay, if the cardiomyocyte is going to grow, it's going to get bigger. There are two ways that it can either get bigger. Either it can get bigger in length, okay, by adding more sarcomeres onto this extended portion up here. So this is our cardiomyocyte here. Okay, now what could happen is you could extend the length and add more sarcomeres on, okay, like so. That is known as eccentric hypertrophy. Okay, now what would be the result for the entire heart of the cardiomyocytes undergoing eccentric hypertrophy? Well, the result of eccentric hypertrophy is that the heart ends up dilating. Okay, and I'll just move this up a little bit. So basically, if you imagine, let me draw a little picture of the heart. Because I'll draw a very simple picture of the heart. Here are our four chambers: the right ventricle, the right atrium, the left atrium, and the left ventricle. Okay, if you imagine what's going to happen if all of the cells in the wall get longer, well, that's going to mean that the circumference of the entire wall is going to become bigger, okay, because remember, these cardiomyocytes in the walls are going to be lined up end to end like so, okay. Now if they're all going to get longer, then if you imagine that there's circles of them going all the way around the heart pretty much, okay, that's going to mean that the entire circumference of the heart gets bigger. So basically what you're going to go over to is having literally a bigger heart, like so, but the walls won't be any thicker basically. Okay, so if I actually add in a bit of thickness here to the walls, okay, like so, the walls aren't going to be any thicker in this new one, okay, so the walls will still be the same thickness, it's just they'll now be on much bigger chambers basically, surrounding much bigger chambers. Okay, so the heart is going to dilate basically if the cardiomyocytes undergo eccentric hypertrophy. Okay, the other form of hypertrophy is where they thicken their width rather than their length. Okay, so I'll show this like so. This is known as concentric hypertrophy. Okay, and this doesn't result in the heart dilating. Instead, it results in the walls of the heart becoming thicker. Okay, and in concentric hypertrophy, you are adding extra sarcomeres on to the width, basically. So all of this new space is going to be new sarcomeres added in there. So it is contractile machinery, basically. Okay, and what's this going to result in? Well, the heart's going to remain the same size, okay, like so. So it's not going to dilate, but now the walls are going to become hugely thickened. Okay, like so. because the cardiomyocytes have all become thicker. So if you imagine them all becoming thicker now, that's going to thicken the walls, basically. Okay, right, so those are the main two forms of hypertrophy of cardiomyocytes: eccentric hypertrophy and concentric hypertrophy. Okay, now we will come on to what drives the two different forms of hypertrophy. Okay, generally in heart failure you get a mixture of both of them. You get eccentric hypertrophy and concentric hypertrophy. Okay, but in certainly in the form of heart failure that we're going to see, you're going to get both of them. Okay, but in certain forms of heart failure you can end up just getting one. Okay, or at least one dominating over the other, but we're going to see both of them occurring together. Okay, right. So let's now talk about how you induce hypertrophy in cardiomyocytes. Okay, so the chronic sympathetic stimulation is going to be involved in stimulating hypertrophy in the cardiomyocytes, and also angiotensin II, chronically stimulating cardiomyocytes, is thought to be important in stimulating the beginning of hypertrophy. Okay, now the mechanisms that underlie the onset of hypertrophy still are not very well understood, but it does seem that chronic sympathetic stimulation and angiotensin II chronic stimulation have a big role in this. Okay, so let me just remind you of the receptors for sympathetic stimulation and angiotensin II stimulation, because this is going to be important when we come on to discuss medications for heart failure later on. Okay, so the sympathetic nervous system, remember, is going to be stimulating beta-1 adrenoreceptors, beta-2 adrenoreceptors, and alpha-1 adrenoreceptors, which are all on the heart. Okay, and although earlier on we said that the ones which were crucial for the physiological function of the sympathetic stimulation on the heart, i.e. the increase in heart rate and the increase in inotropy, were the beta-1 receptors, the beta-2 and the alpha-1 receptors seem to be important in generating the onset of hypertrophy. So it seems to be all three of them that play a role in triggering hypertrophy. Okay, so these are going to trigger hypertrophy. Okay, the cardiomyocytes also have receptors for angiotensin II on their surface, specifically angiotensin II receptor subtype 1. Okay, and these will also stimulate hypertrophy. Okay, right. Now, the other things are also going to help stimulate hypertrophy. So, mean arterial pressure is chronically hypertensive. We've also got raised central venous pressure and pulmonary venous pressure due to the accumulation of additional blood volume. Okay, these are also going to help to stimulate hypertrophy, and let me explain why. Okay, so we'll start off with mean arterial pressure. Okay, so if mean arterial pressure is chronically high, we know what this means. Okay, this means that every time the left ventricle wants to eject blood into the arterial system, it's going to have to build up the pressure inside here to a level higher than that elevated mean arterial pressure. Okay, so that pressure in there is going to be damaging the walls of the heart, basically. When the left ventricle has to build up that huge pressure because of the chronic hypertension, that's going to be damaging the wall of the left ventricle. Okay, and it's going to be occurring the same for the right ventricle, because remember, because the left ventricle's stroke volume is reduced, the pulmonary arterial pressure ends up being too high as well. So the right ventricle is going to have to be building up pressure inside it to a higher pressure than is physiological, okay, and therefore is going to be getting damaged by that as well. So because the afterload is so much higher, and because you have to build it up to that high pressure, it's going to be damaging the wall of the heart, okay, and this is what's known as pressure overload. Okay, so because the ventricles are fighting against too high arterial pressures, okay, they can get damaged by that very high pressure that they're having to build the blood up to actually get blood to eject into the arterial systems. And pressure overload is important in driving hypertrophy. Okay, Right, now on to why central venous pressure and pulmonary venous pressure being too high is going to drive hypertrophy. Well, they cause what is known as volume overload. Okay, so remember, when the heart relaxes, it firstly relaxes to its natural volume and then fills up with blood, and then because of the central venous pressure and pulmonary venous pressure, you get a little bit of extra blood squeezed in and the heart wall stretches to accommodate that. If these are increased, we've already discussed that that's going to increase that end diastolic volume. It's going to increase the amount of blood that's going to be squeezed in. Okay, and we've discussed that that is slightly beneficial because of the Frank-Starling law. That's going to help to increase stroke volume. However, chronically too high end diastolic volumes leads to chronic stretch on the ventricle walls, okay, which is too high, and that's going to actually damage the ventricles as well. Okay, so ventricle stretch seems to also trigger hypertrophy. Okay, and it's not known what the receptors within the cardiomyocytes for pressure and stretch are basically, but we're confident that some sort of receptor for those exists and that it somehow activates hypertrophy within the cardiomyocytes. Okay, so because volume overload causes stretch in the cardiomyocytes, and because pressure overload causes the pressure on the cardiomyocytes, these two things are also going to drive hypertrophy. Okay, and that's how all these four things gradually lead to the hypertrophy of the cardiomyocytes. So the cardiomyocytes in the ventricles are going to start undergoing this hypertrophy, and you might say, well, why is this a bad thing? This seems like a fantastic thing. Okay, we're going to make a stronger heart. Okay, this should deal with the entire problem. The problem is that nature doesn't seem to have mastered hypertrophy brilliantly. Okay, when the cardiomyocytes start to undergo these hypertrophic processes, initially they do gain strength, but over a long period of time, these hypertrophic cardiomyocytes, they just start to deteriorate basically. Their strength just plummets, okay? It goes up for a while and then it just completely plummets off. Their ability to contract just dies basically. They don't die necessarily, okay? But they just gradually lose their ability to contract, and we will have a look at the mechanisms that are thought to underlie why they lose their ability to contract, okay? Right, so basically it buys you a little bit of time. They do work better initially, but then for some reason, and these reasons aren't understood, we don't know what fundamentally causes the malfunctions that then lead to them losing their contractile ability. We do have a good idea of what has actually gone wrong in them, but we don't know what actually causes those things to go wrong. Okay, but it just seems like nature hasn't fully perfected this, basically. Okay, that it starts this hypertrophic process to try and make the heart stronger, but then it ends up failing, basically. And the cardiomyocytes that hypertrophy like this just start to deteriorate. Their calcium signaling processes just malfunction, basically. Okay, right. The other thing I want to say is what drives eccentric hypertrophy and what drives concentric hypertrophy. Okay, so basically, this is the general rule, okay? It's probably oversimplified, but this seems to be the general rule. Volume overload, okay, so excessive stretch on the cardiomyocytes seems to drive eccentric hypertrophy, okay? So, too high central venous pressure and pulmonary venous pressure, which causes that overloading of the ventricles when they're at the end of diastole. Okay, that seems to drive eccentric hypertrophy, where the cardiomyocytes increase in length more than they increase in width, and therefore a dilated heart. Okay, whereas pressure overload, which is caused by the too high pulmonary arterial pressure and the too high mean arterial pressure, which means that the ventricle has to build up these massive great pressures within them before they can actually eject blood out, that seems to drive concentric hypertrophy. Okay, so we've seen in our hypertension induced cardiac failure, that we're going to have both volume overload and pressure overload, and therefore we're likely to get a mixture of eccentric hypertrophy and concentric hypertrophy. So we will get a dilated heart, but we'll also get thicker walls as well. Okay, and that's the most common thing to happen, that you get a mixture of eccentric hypertrophy and concentric hypertrophy. But in certain cases you can get very extremized cases where it's towards one of these and away from the other. Okay, right. So, what we now want to turn our attention onto is what happens in these hypertrophied cardiomyocytes to cause them to lose their ability to contract. Okay, and it's completely and utterly, we think, about the calcium signaling. Okay, they seem to have huge malfunctions in their calcium signaling processes, and the calcium signaling processes underlie the initiation of contraction by the cardiomyocytes. Okay, so we don't know why calcium signaling malfunctions in these cardiomyocytes that have undergone hypertrophy, but we do know which bits of the calcium signaling have gone wrong. We just don't know why it's happened, basically. Okay, so what I want to do then is I want to firstly go over normal calcium signaling in a cardiomyocyte, and then we'll have a look at what has changed in these hypertrophied cardiomyocytes. Okay, and we'll then have a better understanding of why these cardiomyocytes are then losing their ability to contract. Okay, it will also help us when we come on to discuss some of the heart failure medications to have a good understanding of the calcium signaling within the cardiomyocytes. Okay, but I suppose before I actually go on to discussing that, we should actually just say how this thing ends. Okay, so basically we've now discussed that because of the neurohormonal input, this is the chronic situation, that you've got chronic SNS input, you've got chronically high mean arterial pressure, you've got chronically high central venous pressure and pulmonary venous pressure, and you've got chronically high angiotensin II. What this is going to lead to is it's going to drive the cardiomyocytes to hypertrophy, and then gradually these cardiomyocytes are just going to start completely losing their ability to contract. So over time what's going to happen is the heart is going to become weaker and weaker and weaker as more cardiomyocytes start to just lose their ability to contract, basically, and become weak and feeble, and it gets worse than it was initially. Okay, so the heart becomes weaker and weaker and weaker, and then it gets to the point that cardiac output just falls more and more and more and more, and even with chronic sympathetic nervous system stimulation, the heart's cardiac output can't really be raised anymore. Okay, and then when cardiac output falls to the level that it's no longer sufficient to meet the needs of the peripheral tissues, even when you're shunting as much blood as possible to the brain and the heart, then of course it's going to be fatal. Okay, and that's how heart failure propagates, basically, how it gets worse with time. It's because of this chronic these chronic conditions which drive the hypertrophy, which then isn't perfect, unfortunately. In fact, it's not nowhere near perfect. Okay, nature hasn't perfected it, and the cardiomyocytes temporarily get stronger, but then deteriorate for reasons that aren't very well understood. Okay, but it just seems that nature hasn't really perfected this adaptation. Okay, right. So what we want to do then is discuss normal calcium signaling within cardiomyocytes. Okay, so firstly then, let's draw a cardiomyocyte. Okay, so I'll draw, in fact I think I'll go onto a clean sheet of paper for this, because this is going to be quite an important drawing and I don't want to have to continuously be flipping back and forth. Okay, so let's draw a cardiomyocyte here. So remember, cardiomyocytes have these invaginations of their cell membranes known as T-tubules, or at least if we're talking about a ventricular cardiomyocyte, they do. The atrial cardiomyocytes don't have them. Okay, but of course we've been talking about ventricular cardiomyocytes the entire video, so we're not going to change that suddenly now. Okay, so here are a few T-tubules shown here. Okay, so this is our cardiomyocyte. So they've got these finger-like invaginations into their cell membranes, which invaginate into the cytoplasm. Okay, the other important thing to put on the cardiomyocyte right away is the Sarcoplasmic Reticulum, which is a massive, great intracellular organelle that's going to go all over the place, basically. So I'll put this here. Now, it's not got one continuous lump like that, it's more like a network, basically, so I'll put some holes in this to make it look more realistic. So it's a great sort of tubular network, like so. Okay, and I'll colour it in a moment to get the point across. Right, that does it. Okay, so this great tube here, this is the Sarcoplasmic Reticulum, and it's a great sort of network of tubes that are all sort of joined together. And this is an intracellular store of calcium. Okay, right. So the other important thing to discuss is what are known as dyadic junctions, which are an example of something known as a calcium synapse. Okay, so basically the Sarcoplasmic Reticulum, which I should have labelled up here, this is the Sarcoplasmic Reticulum, the equivalent of the endoplasmic reticulum, but because we're talking about a muscle cell, we put the prefix sarco rather than endo. Okay. Now, off the Sarcoplasmic Reticulum, we're going to have little processes which are going to form these special sort of synapses with the cell membrane. Okay, now the cell membrane also has a special name when we're talking about muscle cells. Okay, it's known as the sarcolemma. Okay, so basically you have these junctions between the sarcolemma here. Okay, and I'll highlight this up. So here's the sarcolemma, here in turquoise. Okay, and then you have a junction between that and the Sarcoplasmic Reticulum membrane here in orange. So you have that little process coming off the Sarcoplasmic Reticulum here in blue. Okay, and that's going to have an interface basically with the sarcolemmal membrane here. And this junction between the sarcolemmal membrane and the Sarcoplasmic Reticulum membrane, this is known as dyadic junction. Okay, so this is called a dyadic junction, and it's an example of a calcium synapse, which is a much broader concept that you find in all sorts of other cells. So this is called a calcium synapse, and it's called that because of its analogies to the synapses between neurons. Okay, so remember, in synapses between neurons, in the classical chemical synapse between neurons, what happens is the presynaptic neuron releases a neurotransmitter which then works on the membrane of the postsynaptic neuron. Okay, well, this is actually going to operate in a very similar way. This sarcolemmal membrane here is analogous to the presynaptic membrane, and then the Sarcoplasmic Reticulum membrane here is analogous to the postsynaptic membrane, and the little bit of cytoplasm in between, and there's another fancy word here. So rather than call it cytoplasm in muscle cells, it's known as myoplasm. Okay, so that little bit of myoplasm in between the two membranes is analogous to the synaptic cleft, and in fact is called the dyadic cleft. Okay, right. So, these are going to be important in triggering contraction, as we'll see in a moment. Okay, right. Now, before I talk about how you're actually going to generate the calcium signal that causes contraction, okay, what I firstly want to talk about is normal calcium homeostasis in these cardiomyocytes when they're not in the process of contracting. Okay, so when they're not having a calcium spike. Okay, so let's start then with the cardiomyocyte at rest. So let's imagine that it's not receiving any action potentials and therefore it's not being triggered to contract. What is the normal status of calcium signaling in this cardiomyocyte? Well, the normal status of calcium signaling in this cardiomyocyte is that calcium level in the cytoplasm is at around 10 nanomolar. Okay, so very, very low. Contrast that to the extracellular calcium concentration, which is around 1.5 millimolar. Okay, so that's an 150,000-fold gradient of calcium across the cell membrane. Okay, so it's a big, big gradient of calcium across the cell membrane. Okay, right. You then also have much higher calcium concentration in the Sarcoplasmic Reticulum. Okay, so how do you keep a constant calcium concentration in the cytoplasm then? Okay, so let's draw a little picture here. So we'll say this is the sarcoplasmic reticulum here, we'll say this is the sarcolemma. So let's colour code this. So this is the sarcolemma here in turquoise. Okay, here is our sarcoplasmic reticulum here in blue. Okay, so we're saying that calcium is very, very high extracellularly. It's also quite high in the sarcoplasmic reticulum here, but it's very low in the cytoplasm. Okay, so how is this maintained? What's the normal equilibrium? Well, let me tell you now that there is a leak of calcium back into the cytoplasm from the extracellular fluid. Okay, so there is a leak across the sarcolemma into the cytoplasm all the time. Okay, so some calcium ions leak back into the cytoplasm all the time. Okay, and there is also a leak out of the sarcoplasmic reticulum. Now, as far as I'm aware, the origins of these leaks are not yet known, i.e. which proteins are actually involved in allowing these calcium ions to move across the cell membrane. It's not known. Okay, it's very unlikely that they just go through the cell membrane on their own, okay, because they are quite a polar ion, and therefore it's unlikely that they're going to be able to get through a phospholipid by there. Okay, but there is calcium leaking out of the SR lumen and also calcium leaking into the cytoplasm from the extracellular fluid. Okay, so if we're going to maintain a constant cytoplasmic calcium concentration of 10 nanomolar, then we better make sure that we chuck back out the calcium that's coming in from the leak from the extracellular fluid, and that we put back in the calcium that's coming out of the leak of the sarcoplasmic reticulum here. Okay, so how do we do that? Well, let me start with how we get the calcium that is leaking out of the sarcoplasmic reticulum back into the sarcoplasmic reticulum. Okay, so basically in the sarcoplasmic reticulum membrane, you have a special pump. Okay, and this special pump is known as Circa 2A. Okay, so there are many different forms of Circa. The one in cardiomyocytes is specifically Circa 2A. Okay, so I think I'll colour in Circa 2A in red here. So this is Circa 2A. So what does Circa stand for? Circa stands for the sarco, that's the S, and then it's / endo, and then it's endoplasmic. So S is for sarco, E is for endoplasmic, R is for reticulum, and then the Ca is for calcium ATPase. So it's the sarco/endoplasmic calcium ATPase, and then it's specifically the sarco/endoplasmic calcium ATPase 2A. Okay, right. So what does the Circa 2A usually do? Well, it's basically an active transporter that is going to return calcium into the cytoplasm of the sarcoplasmic reticulum. Okay, so what it's going to do is it's going to couple the movement of calcium ions back into the lumen of the sarcoplasmic reticulum to the hydrolysis of adenosine triphosphate. So one molecule of ATP is going to be hydrolyzed to ADP and an inorganic phosphate, and for that energy, what you're going to do is you're going to pump 2 calciums back into the lumen of the sarcoplasmic reticulum, but you're also going to bring stuff out of the sarcoplasmic reticulum lumen. You're going to bring 2 protons back out as well. Okay, so basically the Circa 2A is a primary active transporter that couples the movement of calcium into the endoplasm or sarcoplasmic reticulum lumen to the hydrolysis of ATP. So the hydrolysis of ATP releases the energy that is needed to move the calcium into the lumen of the sarcoplasmic reticulum, i.e. up its electrochemical gradient. Okay, right. So that's how you balance this leak. So at equilibrium, you're going to have an equal amount of calcium leaking out as is being replaced into the sarcoplasmic reticulum by the Circa 2A. Okay, right. You also then need to balance the leak across the plasma membrane. Okay, and this is done through 2 separate mechanisms. Okay, so I will introduce the most important one first, but the most important one, unfortunately, is more complicated to understand because rather than being a primary active transporter, which are very easy to understand, it is a secondary active transporter. Okay, so this one here is the NCX1. Okay, now what does this stand for? Well, first the N stands for Na, for sodium. Okay, the C stands for calcium. Okay, the X stands for exchanger, and it's the sodium-calcium exchanger 1. Okay, right. So I'll colour in the sodium-calcium exchanger 1 in green here. Okay, so what the sodium-calcium exchanger does is it allows one sodium into the cytoplasm of the cell in exchange for one calcium ion being chucked out. Okay, now sodium's electrochemical gradient favours its movement into the cell. Okay, so sodium concentration is much higher extracellularly than it is intracellularly. Okay, so the concentration gradient favours the movement of sodium into the cell. You also have to take into account the electrical gradient. Okay, the electrical gradient will most definitely be negative. Okay, so negative 70 or even more hyperpolarized in a cardiomyocyte. Okay, so that will mean that the electrical potential will be lower intracellularly than it will be extracellularly. Sodium is a positively charged ion and therefore wants to go where the electrical potential is lower, which will be the intracellular compartment. Okay, so sodium is moving down its electrochemical gradient when it comes into the cell. Okay, that will release the energy that is then used to move the calcium out. Okay, so this is an example of a secondary active transporter because it is coupling the movement of calcium up its electrochemical gradient to the movement of another ion down its electrochemical gradient, rather than coupling it to a primary chemical reaction itself. Okay, then the other mechanism for extruding calcium is the PMCA, okay, which stands for the plasma membrane calcium ATPase. Okay, so the P is for plasma, the M is for membrane, okay, and then the Ca is for calcium ATPase. Okay, now this is an active transporter that is a primary active transporter. Okay, so I will colour in PMCA in vivid purple here. Okay, so this is going to chuck one calcium ion out of the cytoplasm of the cell, or rather the myoplasm. Okay, so one calcium is going to leave, and every time it does that, it's going to hydrolyze a molecule of ATP, which I'll just put in here. So ATP is going to be hydrolyzed to ADP and inorganic phosphate. Okay, right, and that's how you compensate for this leak of calcium into the cytoplasm of the cell by extruding the same amount of calcium as is leaking in via the plasma membrane calcium ATPase and the sodium calcium exchanger 1. Okay, so at equilibrium, this is how you're maintaining a constant cytoplasmic calcium concentration, by having these leaks out of the SR and into the cytoplasm from the extracellular fluid, compensated for by these counter mechanisms which are returning it back out of the cell and pumping it back into the sarcoplasmic reticulum. Okay, and it's very important to understand these. Later on we're going to need to understand these very well. Okay, right. So, now let's talk about how you cause the calcium rise throughout the cytoplasm of the cardiomyocyte in order to trigger contraction of the cardiomyocyte. Okay, so contraction of the cardiomyocyte is triggered by cytoplasmic calcium concentration rising from around 10 nanomolar to around 1 micromolar. So it rises by a factor of around 100, and when the calcium goes up to that level, that's what triggers the cardiomyocyte to contract. Now, we're not going to go into the detail of how the calcium causes it to contract. We don't need to know how it contracts to understand what goes wrong in the hypertrophied cardiomyocytes and to understand how some of these medications for heart failure work. Okay, so we're just going to leave the story there at when the calcium rises from 10 nanomolar to 1 micromolar in the myoplasm, that's what causes the cardiomyocyte to contract. And we're going to now look at how does an action potential propagating along the membrane of the cardiomyocyte, the sarcolemma, trigger calcium to rise from 10 nanomolar to 1 micromolar. Okay, so basically what is going to happen is the action potential is going to propagate along the sarcolemma. okay? And it's going to propagate along all of these T tubules, and you'll have loads of these all over the cardiomyocyte, okay? And when it propagates down at these T tubules, it's going to come into contact with these Diavik junctions here that are all over the T tubules. You also do have a few Diavik junctions with the actual sarcolemma that's not in the T tubules. Okay, so here's a Diavik junction onto a bit of sarcolemma that isn't a T tubule. Okay, and what's then going to happen is when the action potential reaches the sarcolemma membrane in these Diavik junctions, it's going to trigger a little bit of calcium to come across the sarcolemma into the Diavik cleft, and that's going to activate receptors for calcium that are in the membrane of the Sarcoplasmic Reticulum in these Diavik junctions. So let me draw this larger. Okay, right. So here is this little process coming off the Sarcoplasmic Reticulum here. Okay, this is the Diavik cleft here. This is the sarcolemma. Okay, so let's just colour in these portions. I'll just raise this a little bit. So this in turquoise here, this is the sarcolemma. Okay, whether it's a T-tubule portion of sarcolemma or just a surface portion of sarcolemma. And here, this is the Sarcoplasmic Reticulum little process. Okay, and then we've got the Diavik junction here with this space in between, known as the Diavik cleft. Okay, right. So basically, when the action potential propagates along the sarcolemma, what of course is going to happen is the electrical potential difference across the cell membrane is going to be depolarized, okay, and that's going to actually activate special voltage-gated calcium channels in the membrane of the sarcolemma, okay, here. And these are what are known as L-type voltage-gated calcium channels, which is a special type of voltage-gated calcium channel. And for short, I'm going to abbreviate voltage-gated calcium channel down to VGCC. I suppose I should probably write it out in full just once. So VGCC is my abbreviation for voltage-gated calcium channel. Okay, right. Now, the L-type voltage-gated calcium channels are frequently known by another name. Okay, they're also known as the Dihydropyridine receptors. It's referring to the exact same thing, or DHPR receptors. So this stands for Dihydropyridine receptors. And this just refers to the fact that there is a massive, great collection of drugs known as dihydropyridines, okay, which are capable of blocking these receptors. And since these receptors are susceptible to dihydropyridines, some people have named them after the drug the drug class which blocks them, and therefore that's why they're known as the dihydropyridine receptors. So whichever one you want to use, you will see them both used frequently. So dihydropyridine receptors, or L-type voltage-gated calcium channels. The important thing to understand is that when the electrical potential difference across the cell membrane becomes depolarized, such as in an action potential, these things are going to open, and they're going to allow calcium to move from the extracellular fluid into the cytoplasm, because remember, there is a massive, great concentration gradient of calcium across this cell membrane. Okay, so when you open a calcium channel, there's no question of which way the calcium is going to move. It's going to move into the cytoplasm of the cell. Okay, so you get a little rise in calcium in the Diavik cleft. And this little rise in calcium is so important that it's been given its own name. Okay, so it's called a calcium sparklet. Okay, so when calcium goes up in the Diavik cleft because of the L-type voltage-gated calcium channels in the sarcolemma opening, that's called a calcium sparklet. Okay, right, and of course you're not just going to have one L-type voltage-gated calcium channel in this Diavik junction, you're going to have multiple of them in here. Okay, and the calcium sparklet is then going to activate receptors that respond to calcium that are in the Sarcoplasmic Reticulum membrane. Okay, so I'll put this here. So in the Sarcoplasmic Reticulum membrane, you have receptors known as type 2 ryanodine receptors. Okay, for short, abbreviated down to RYR2. Okay, so this stands for type 2, and then the RY is for ryanodine receptors. Okay, so the RY is for ryanodine, and then the R is for receptors. Okay, so this is a type 2 ranitidine receptor, or an RYR2 here. So I'll colour that in light green here. Okay, right. Now, type 2 ranitidine receptors are calcium channels themselves, but they are opened by calcium. So the calcium going up in the diatic cleft is actually going to open the type 2 ranitidine receptors, and they're then going to release calcium from the sarcoplasmic reticulum into the diatic cleft, and they release much more calcium than the L-type voltage-gated calcium channels allow into the diatic cleft. Okay, so now you release more calcium. Okay, and the calcium released from the type 2 ranitidine receptors, the rise in calcium in the diatic cleft due to the opening of the type 2 ranitidine receptors, that is called a calcium spark. Okay, so calcium sparklets and calcium sparks there. Okay, right. And this release of calcium from the type 2 ranitidine receptors hugely amplifies the rise in intracellular calcium concentration, and this happening all over the cell. Okay, so you're going to have loads of these diatic junctions all over the cell, and they're all going to be releasing calcium into the myoplasm all at once. This massive effect is what's going to trigger the rise in intracellular calcium concentration from 10 nanomolar to 1 micromolar. Okay, right. So those are the type 2 ranitidine receptors. Okay, right. And by the way, this release of calcium from the type 2 ranitidine receptors that is triggered by calcium has a special name. It's known as calcium-induced calcium release. Okay, let me just make sure this is in the screen. Calcium-induced calcium release. And for short, calcium-induced calcium release is often abbreviated down to CICR, CI for calcium-induced, and then CR for calcium release. Okay, right. So you get this calcium-induced calcium release occurring at these diatic junctions, and this is why it's compared to a synaptic junction, because the sarcolemma is effectively releasing its calcium, which is analogous to a neurotransmitter, okay, which is then acting on receptors on the membrane of the sarcoplasmic reticulum membrane, okay, and that then is triggering a response in this sarcoplasmic reticulum. Okay, right. So now what we want to discuss is how relaxation occurs. So now you've got the massive great calcium spike in the cytoplasm of the cell. So calcium has gone up hugely, that causes contraction. What we now want to look at is how do you reduce the calcium back down in the cytoplasm of the cell in order to relax the cardiomyocyte. Okay, because it's the calcium going down that relaxes the cardiomyocyte. Okay, so firstly, when calcium builds up to a high enough level in the diatic cleft, it inactivates the L-type voltage-gated calcium channels and the type 2 ranitidine receptors. So it forces these shut, basically, by mechanisms that are still not very well understood, but it causes them to shut. Okay, so you stop letting in more calcium from the extracellular fluid, and you also stop releasing calcium from the sarcoplasmic reticulum. Okay, and this will be happening all over the cell. The diatic junctions will be shutting down, basically. So now what needs to happen is we need to remove the calcium that is now within the myoplasm. Well, basically, it is these mechanisms here that are going to now extrude the calcium from the cytoplasm. Okay, so Circa 2A is going to return calcium into the sarcoplasmic reticulum lumen, okay, and about 75% of the calcium that's released in a calcium spike is going to be returned back into the sarcoplasmic reticulum by Circa 2A. The other 25% is mainly going to be extruded by sodium-calcium exchangers, okay, rather than the plasma membrane calcium ATPase. PMCA is extremely slow, basically. So most of the calcium that is extruded out of the cell goes via NCX1. Okay, PMCA is important in the equilibrium, but it's not really important in returning us back down to the equilibrium from the spike. Okay, and once you've then extruded the excessive calcium, what will then happen is you'll re-find an equilibrium where you have 10 nanomolar calcium in the cytoplasm and then everything will be in equilibrium. Okay, right, so we'll continue this discussion in the next video.